welcome everyone to this MIV seminar. So here, Matthias is not coming today, so I'm gonna take the lead to introduce Seung from our lab, who is, we all know Seung. He's fifth year. Yeah, let's give it. Yeah, so he's fifth year PhD students in mechanical engineering department in TAM division, right? So this is also part of your required presentation to give as part of the degree program. So we'll hear something about computing. So it's gonna be really nice. So look forward to the presentation.
Okay, thank you, Xiaotian, for introducing. Okay, so mic is working okay? Okay, so my name is Seung Kim. My advisor is Mattia Gazzola, and here I would like to present my ongoing work for the last 3 years, 4 years maybe, on how to use biological neural substrate to do some computing. The video that you're seeing here is one of the calcium imaging video of the actual living neuron. Here, the green lights, the intensity of the green light is signifying the activations of these neurons. And then you can see there are a lot of like green dots. That's actually the nucleus of these neurons. And you can also pretty clearly see all the connections that they're formed. And you can see some locations are locally synchronized, some locations are not synchronized, and they sometimes just globally light up. So these are a very complicated dynamical system that we're trying to see if there's any way to engineer the system, if you can build any computing device, or if you can develop any further algorithms that's motivated from this kind of system. So in vitro means we are growing the cells outside of the body, and that actually gives us a lot of opportunity to play around with this system. Otherwise, it's going to be impossible if it was actually in the body. And we are trying to see if we can actually play around with this. So instead of looking at the video, there's actually a better way to translate their activities in terms of to understand their dynamics. And this is our usual representation of how to do that. This is a recording from the 128 electrodes, and those are represented in each channels. This is the recordings windows of about 30 seconds. And each of the dots that you're seeing is what we call the spike activities. So spike is individual like neuron activity that we record the timing of when the spike event happened. And they are roughly about 1 millisecond in time. And you can see that there's some local activities going on. A lot of spikes are aligned in multiple channels. Sometimes you see like this global activity, global population activities as well, that we call it avalanche. It's usually about 500 milliseconds to up to like 3-4 seconds. So this kind of activity really resides in like a second range. And then other than that, you also see a lot of like different temporal scale, different spatial scale behaviors. And you can also see that there's some refractory period that happens right after the global activities. So those are also another signatures of the biological neural system. And one thing to remember is this activity is recorded from the spontaneous activities of the neuron. So unlike any artificial neural network, which you need to actually give an input to see the response, this biological system is just continuously living. So they just continuously fire and they have their own activities. And it's somewhat unclear even right now that we actually don't know how to interpret this yet. But one thing for sure is it is just fundamental nature of the system. Just like how our brain just never stops, like even when we are sleeping, it's constantly doing something. And this is like one of the very typical cultures that we make. We grow these 2D motor neuron cultures in the dish on the MEA so that we can record the activities. But I would say if we look at like all different variety of samples that we record over the last 3 years, the amount of variability that we get is quite impressive. Again, all of these samples is recorded from the same type of neuron, same type of density, same type of protocol. I mean, experimentalists do try different things, so there might be some difference. But it is still very clear that the amount of sensitivity and uncertainty that goes into this kind of system is actually quite impressive. So on top of that, they continuously change. So they're living beings, so they age, they get tired, they die after a while. So accounting all that, it's actually very challenging just to start, like how to even approach building some robust system out of this kind of activities. So the amount of recordings that we did over 3 years of period, thanks to Andrew, Kimia, and Gaurav, they were the one providing me all the samples to record and play with. We were able to get quite a reasonable amount of understanding of what their natural behavior is, but they're still restricted to only the motor neuron type. So there's still a lot of room that we need to explore to fully understand what's happening. But this variety really reminds me that one can always argue as an engineer, we can always try to improve the understanding of the system, see exactly how this thing is working by improving the hardwares, by improving the protocols to make the system. But it is, on the other hand, somewhat true that any biological living being will be different from one another. There's always this aleatoric uncertainties in there that even the same twin with the same DNA, with the same family will be a different person at the end, right? So this sort of a variation is probably something we just need to figure out how to utilize it. And here the whole goal of my talk is basically thinking about how to robustly build computational framework across all these diversities in mind. So there's a one quite decades old computational framework called reservoir computing. We usually call RC. And it's originally inspired by the brain network, but nowadays people normally understand it as like a recurrent neural network or LSTM network. But this like old idea specifically addressed this kind of challenges pretty well. So when we have like a variability in the network, but still want to drive the same functionality, reservoir computing gives some nice framework to deliver that. Just like probably if we give some anatomical descriptions of all the neurons in the body, everyone here is going to have a different neural system one to the other, but everyone still be able to talk the same language, can still walk, can still have the same functionality. So those are the kind of system that we're trying to build here. Here I will briefly introduce the concept and how we implement it in our system. So reservoir computing usually consists of 3 different components: input, reservoir, and the output. And the Maas and Jaeger is the one that independently came up with this idea. And the reservoir, the internal system, is the one that corresponds to the biological cultures that we have. It's usually used with some complex and nonlinear dynamical systems. And input is typically used with some continuous signals, although there's nothing stopping us from using the discrete signal as well. And the output is extracted from some subsamples of the activities from different locations of this dynamical system. And the key idea of reservoir computing is that we typically don't train don't really concern what is happening in the reservoir itself. We just let it be some rich dynamical system, and then we only train the output with some linear or ridge regression, and then we try to drive some functionality out of it. So that really saves us from doing some expensive backpropagation. But on top of that, the fact that this middle dynamic, the reservoir can be just any dynamical system, is actually addressing the challenge that I just mentioned. So this network can be just replaced with any other network. So even the reservoir is just completely different structure from one to the other. As long as this reservoir still represents some rich dynamics to encode all the inputs, then we should still be able to train some output to come up with some computational functions. So in our lab, we have this MIV platform, thanks to Xiaotian, who was the one originally leading this project, that we set our neural cultures and we can read the electrical signals with it. We typically use the light as an input source. We have this motor neurons on MEA that's transfected optogenetic to have an optogenetic features, so that when we shine the light with some specific light frequency, they generate some spikes. And then we use the MEA, microelectrode array, to read the activities. And then with some signal processing, post-processing, we can detect the spikes, and we translate this into the firing rate, and then we can do further stuff. So here, usually the theory of the reservoir computing is complete enough to say that this thing, as long as we have a big enough reservoir, it can actually converge into any universal function approximator. Although it is true that not all dynamical system can do the job. There's actually some preferable dynamical system or dynamical characteristic that we want to see in the culture. So that's something we need to check in the middle. So here we, throughout the experiment, we developed some method to do some diagnostics. We call it pre-flight check. The idea is, because we are trying to represent our culture as a reservoir, there's a lot of literatures in the back suggesting like what kind of metrics we can measure to actually see what's the quality of this reservoir. And our hypothesis was that if we can actually get a good metrics on these qualities, then maybe at the end, when we try to use this culture for some computational purpose, it will also do better than the rest. This was specifically useful because we have a very large sample variability. So even among the same batch, usually sample variability is very huge. Some have a high activity over the other. Some have a like high burst rate than over the other. So it was somewhat necessary to sort of understand or filter out what kind of feature is actually what we want. And so that we can kind of early decide and discard the sample that we might not want to use. So over about thousands of samples of recording over 3-year period, we kind of saw like 4 major type of activities over all the motor neuron cultures that we saw. There's actually a 5th type called X. I call type X, that is just so wild that I don't even want to consider it as a good sample. But usually, most of the sample falls into these 4 categories. From the top, we have a very asynchronous behavior. We usually don't see any bursts forming. Also, it's only 7% of the sample show this. So it's usually a sample that's about to die or when they don't form enough connections. Or it also, we see this in like some low density cultures. So that's one of the dynamics that we see. And then type B and C is the one that we see a lot of like intermediate firings. We see some burst synchronization. The difference between B and C is really just the time scale, how fast the burst is happening or how wide the burst is. And the type D is when the activity is just fully synchronized. We don't see any like independent firing that's outside of this avalanche behavior. And we see a lot of samples for type D as well, because if you have like excess amount of neurons in there, and if they're too strongly coupled, at least that theory suggests that they should just form single avalanche behavior without any inhibitory neurons. So the way we classify them is we try to follow this protocol out of empirical reason. So first we load the sample, and then we first check if the sample has a good MEA, if there's any broken if there's too many channels with broken channels with bad impedance, then we typically just discard the sample, because recording and processing is quite expensive, that we typically try to use the good samples as much as possible. We let it sit for 30 minutes. This is sort of a personal reason, that personal experience, that for about first 20 minutes, the firing rate changed quite radically. They decay very fast, and we think it's because during the handling of this sample, we're actually perturbing the sample quite a lot, to the point that when you just let it still and let it rest, it kind of settles to some more steady state behaviors. And then we record about 30 minutes of spontaneous recordings, and then that gives us how to access the type of this sample. We also noticed that once we assess the type, it usually lasts about 30 minutes to 1 hour. And after that, if we just let it sit in the incubator, it starts to actually change. So there's a lot of like aging maturations, maybe they get tired. There's a lot of like changing in the dynamics that's also happening on the side that we are not fully characterizing it at this point. So again, like we usually don't see any synchronized activity. B and C, we choose 0.5 hertz to split the B and C for empirical reason. later on, this rate of burst relates to how fast we can use the system to encode the information. So you can think of it as like a clock rate for the CPU. If the dynamics is running too slow, then the issue is by the time we finish the training, the culture might have just changed enough to the point that we cannot use the collected data. So that's why we naturally want some like fast pace sample. And then usually sample D is pretty clear. They usually just converge into single avalanche behavior. So we can knock those out as well. So next, based on these 4 categories, there are several metrics that we actually measure before we select or before we try to guess which one will work the best. Here, a lot of literatures come from criticality literatures, reservoir computing, and recurrent neural network literatures. There's some comes from like information theory as well. Here, I will just introduce 3 of them. So first is the criticality. So there's a wide literatures in the field, in the neuroscience, that brain, if the brain's trying to work in an optimal way, it needs to sit on some critical regime where the brain is not too chaotic, but not too ordered. So that's the criticality theory. And then the metric that I'm showing here specifically is the branching ratio. And usually the branching ratio of around 1 is when the critical system is, when the system is actually critical. The A is not measured because you actually need an avalanche behavior to actually measure this number. So if you don't have any avalanche, then it's not the metric that you can actually measure. Here, I'm not trying to really say that any of our culture is actually critical, because actually telling whether a system is critical or not, it's not something you can just tell by just a 1 metric for the branching ratio. You actually need to go through a lot of different tests as well. We typically see that our culture is not critical, but at least this branching ratio tells us that the activity that we get in terms of like increasing the activity with respect to the previous activity and how they decay down is at least balanced when for the sample B and C. Sample D, like we get much lower branching ratio because if you see the burst shape, they're very just sharp and very thin. And I think it ultimately relates to how the burst actually looks like. The second metric that we call kernel rank comes from the reservoir computing literature. So you can think of the reservoir as the information encoding layer. So if the encoding layer to work optimally in this theory, the information needs to be mapped into higher dimensional feature space. And what kernel rank tells us is that it's the rank of the dimension in the feature space. So the larger the number is, the reservoir should be able to work better. The issue with the kernel rank is that if we try to only increase the kernel rank higher, then we tend to push the system to be more dynamical system to be more chaotic at the end. So there's another metric called generalization rank. So to measure this, the idea is that when you give a similar patterns to the system, the result or the behavior should also be similar to each other. So it's very closely related to the chaoticity of the system. So if the system is too chaotic, if two very similar input becomes a very different response, then it might be able to represent very large features in there, but it's going to be hardly usable because we won't be able to decode any information out of it. So based on this 3, and there's actually more metrics, but we can tell that sample C and D is roughly a good candidate for our purpose. B is typically performed. A and B is we typically don't really use it for further experiment. And then the last diagnostic that we do is we try to find some space embedding trajectories. So the idea is that when we get this high dimensional vector of the firing rate informations, then there's a hypothesis that the actual internal information that it's trying to represent is sitting in the much lower dimension. So there's a lot of existing method like a Gaussian process vector analysis that tries to interpret what this low dimensional trajectory is. And this gives us some nice visualizations of our the neural activities, but it also, we can use it for, we can use it to interpret the behaviors. We can use it to see if if the dynamics is changing over time, and we are currently trying to see if we can make any further applications out of this, this line of work. This is a static picture of the dynamic representation for 4 different types. So the type B and C, I just added one picture because they actually look roughly the same, just with a different time scale. If you look at the type A behavior, you can see the trajectories are mostly clumped at the center. It means that there's really no dominant behavior that's represented in the system. But when you see a type D, they have a very uniform burst behavior. So the trajectory also looks very uniform and circular. So you can see the attractor state and oscillating behavior of this burst. And type C and B is where you see much more rich dynamics. They do have a representative features, but they also reside in much wide volumes of the space. So we suspect that, or we hypothesize that those are more optimal dynamics that we want to see to encode like more informations in the reservoir. So now we have some metrics that we measure for different types of behaviors. Now we want to set a very simple benchmark to see if this pre-flight checks is actually telling us anything useful. So here, for the experimental details, we use a light pulse and we encode the patterns in the frequency domain. So the idea is for different patterns from pattern 0 to 9, we give a different number of pulses in fixed bin size or fixed pattern size, which is about 1 second. And then at the end of the pattern, we measure the firing rate and try to construct the state vectors. And we train the reservoir for about 1 hour. So because the pattern size is 1 second, we can collect about 3,600 samples. That's usually enough to train some model with 10 different patterns to classify. So, and then we use a ridge classification, like how the RC is working, to get the linear map. And then later on during the test period, we can measure the reconstruction score or the accuracy to assess the sample. So unlike other like classification for deep learning or any other classical classification problem, the difficulty here is that we cannot do mini batch of the data, right? Like because the biological system needs to process the information sequentially at this point. So we need to push in the pattern one at a time for 1 second. So that gives us a little bit of limitations of how much we can train this sample before they actually change the behavior. So we found that 1 hour is about the optimal time that we can actually use. And the way we test is we give some rest, and then we see if this trained model still can do some classification job after the rest. So the idea is that if the culture is changing over time, we want to see that our model can still be used after that amount of time. So this is the result. So the left plot, I'm trying to only show the first 1 hour of the accuracy. So if we just do random guess of the 10 different patterns, then you will get about 0.1 or 10% chance. That's what the bottom line is about. And the 1 means you'll get 100% accuracy. So you can see that type C and D, the blue and green, does pretty good. And they also somewhat maintains its level for first 1.5 hours. And then type B does slightly better than type A, but the type A really gets the score. It cannot get the score above 50%. The black dots on the top represent the upper bound. So that's actually the best performing sample that we get. I just want to show you that we do get about 90% test score once in a while. But here we want to deliver a little bit more robust methodology overall. So we're looking at the average of the behavior. So all this data is collected over more than 200 samples. So I think it's a good preliminary result to see what's the difference between the dynamics that's going to perform in the RC. So let's just focus on the type C here because that's one of the best performing dynamics that we see. When it can reasonably distinguish the pattern, when it's around like 80% accuracy, the trajectory looks much more consistent like that. So that's the 1 second trajectory evoked by all the inputs. And that's each of this line is an average of 20 different patterns. And as you can see, like different digit can be recognized because the trajectory is quite well separated from one another. And then by the time they go down all the way to like about 30%, so after 4 hours, you can see that trajectory also collapsed down. So it's the way I interpret this is the dynamical behavior and the spontaneous activity does change quite significantly over time. And then whatever the train that we did previously will no longer be valid to estimate after this much of a rest. It usually doesn't fall all the way down to 10% because the amount of confusion happened a little bit imbalanced between low frequency pattern and high frequency pattern. So over time, like it gets more difficult to recognize difference between the high frequency, like pattern 8 and 9, versus pattern 1 and 2. Because even after like 5, 6 hours, it might be able to still distinguish pattern 0 and 1 because the difference in the firing rate will be much greater than 8 and 9. So just water quick. Now, so that was our preliminary result, how the accuracy score changed, how the RC benchmark score changed over time for different type of dynamics. And now our next goal is, can we do anything to really improve this result? So the idea is, can we make any effort to sustain this dynamics for a longer period of time? And we wanted to increase the consistency of the behavior, and it's gonna make us be able to use the system more longer, so that we don't have to just do training for 1 hour, use it for another 1 hour, and then this sample will be gone. So we borrowed the idea from the chaos control theory back in the 90s. So there are a lot of theories back then to stabilize a dynamical system, and a lot of technology has been evolved into making pacemakers or building some stability control. And then recently, it also shown that a lot of the method also work in the stochastical system, including the neural network system. So we wanted to borrow some idea from them to see if we can do any better. So the simplest way that we tried was we add some stabilization by adding the periodic perturbation to the system. So we give a little bit of external input instead of just letting it run spontaneously on its own. And the way we do that is we have another light source on the bottom, and then we add some very low power background modulation to the system. And then we hope that it kind of regulates the dynamics. And so that dynamic is not entirely controlled by the spontaneous, but also we have a little bit of an input. We have a little bit of controlled input that makes system to run a little bit more consistently. So if you look at the trajectory, the difference is quite clear. So you can see that without the modulation, the trajectory is just like what we saw before. It stays in a very large volume, but with the modulation, it kind of collapsed down a little bit. But each of the trajectory looks much more stable. We are probably sacrificing a little bit in the feature space by doing this, but there's a lot of we gain a lot of regulations by doing this. So for example, like so if we actually look at the raster plot, so if you actually look at the spiking activities, without the modulation, the difference is pretty clearly visible. So you can see the burst. The background burst happens on the top without the modulation, regardless of when the pattern starts. But with the modulation, you can see that we can almost always align when the pattern is going to be started with respect to this burst cycle. And when we actually see the timing, the burst interval without the modulation, although we started the sample with the burst frequency about 1 second, we tried to select the sample so that we can keep that consistent. Over time of 1 hour period of training, they still change. They still either drop, in this case, the frequency or the interval decreased, so burst rate actually went up. But we also see a lot of difference changes over time. But with the modulation, you can see that the burst rate just can be fixed by exactly 1 second for entire training period. That's going to allow us to gather much more consistent data. And then at the end, the result for the training is going to be much more generalizable as well. So this is the result from the before without any modulation. Here, let's just focus on the type C and D because they are just two best performing result that we get. And this is with the modulation. You can see the usability of the system is extended much more, all the way up to like 5, 6 to 8 hours for some samples. You can see the trajectory, or you can see the accuracy doesn't decay as fast. And when we actually look at the trajectories, even after 5, 6 hours, the trajectory still remains roughly recognizable, even after 6 hours. So our viewpoints of treating this reservoir as a populations of dynamical system kind of gives us good intuition on how to select a good sample among all the different dynamics that we're going to see. And we can assess the quality. We can see like which one will do better in terms of doing the computation in different tasks. And this is just one of the proof of concept that I've been working on. But I think this example gives a good story of how understanding the dynamics and the state space of how this thing is evolving can actually help building some strategy to improve the system. Now, along the same line of thought of using the state space embedding, there's another idea that we're trying to pursue that called knowledge transplant. So one of the key downside of the whole computing with biological system is that they die. So it's like you can only use your CPU for 1 day, then like it's very hard to make any applications out of it. Because you need to go through all the training process, which takes 1 hour, and then there's very short period of time that you can actually use. Then it's very hard to sustain anything. But that's usually not how the biological system works, right? Like because any intelligent system, we learn each other. Like we can easily transfer the knowledge, or we can easily transfer what we learn from one person to the other. We teach each other, and then that's how we build up the intelligence as a community. And we were trying to think of a similar situations for this neural culture so that even after we finish the training for one sample, we can somehow leverage that training so that we can continue training for the different samples. So let's imagine you have two different cultures. So obviously they're going to be different. They're going to be connected different. The dynamic is going to be different. But then when we shine the input and when we give some patterns that they need to encode, their internal representation would roughly be similar because the input is still similar. So the idea is that maybe one dynamics can be mapped to the other. And as long as if we can easily find this map, then we don't need to go through expensive training process for the student sample that's trying to learn. And so that we can still reuse or leverage some of the information that we learned from the first teacher sample to accelerate the training. So imagine you have an expert and novice, where expert, we went through the full reservoir computing training. It has seen a lot of samples so that we already know how to make the state space embedding. We already found the RC map. And then let's say we want to move on to the second sample, which we haven't observed enough yet to construct the RC map. But then how can we still build the same function using the novice sample? So the idea is that if you can find the lower dimensional state space embedding of how the dynamic is working, then we should be able to find some inverse map of this RC to get roughly the same to do the transplant of this pre-trained knowledge. This is rather possible because a lot of the mapping here is actually linear. The fact that reservoir computing is only dealing with the last layer, the linear training way, is actually a reason why this methodology can work. Yeah, we're trying to see if this can be actually used with the actual biological sample. So I only have a very preliminary result at this point. So the blue curve here is the novice RC. We're doing the RC from the scratch. And then we can apply the algorithm to transplant the idea, the weight from the previously learned sample. That's the green line on top. So if you have any transfer learning background, you can see that like there's some jump bootstrapping effect in the initial point. That's a good sign that there's some information has been transplanted in some meaningful way. But you can kind of see that at the end, they kind of cross over together, and then the blue curve actually goes up. That probably means that sample that I used here, by the time, by like after 10 to 20 minutes after, the dynamic has been changed enough to the point that it might be just better to train from the scratch. So that's somewhat the result that I see, because we have a lot of changes in the dynamics. But I think with the background modulation strategy that we use in the back, we hope that we can improve this result much better in the future. So I will quickly go over some of the roadmaps and some of the things that I've done for this project. I've been like broadly just working in the building systematic method for doing the computing for this neural culture. I participated in working in simulation, some information processing and communicating between the biological culture, the cybernetics. I also do a lot of like robotics problems, and I try to study a lot of like how the biological system learn. And RC plus this dynamical control is like one aspect that I've been working on. I was also involved in developing like a reinforcement learning framework with neuromorphic hardwares. And then I'm trying to work more in the future for how to integrate like different modality of the sensory inputs to in the neuromorphic system. And then I also have a little bit of side projects on converting the artificial neural network to spiking neural network. So if any one of you wants to work with the spiking Loihi kind of neuromorphic system, let me know. And then I also have a lot of interest in the emerging properties of this sort of a neural network system. Yeah. So here's Andrew, Xiaotian, Gaurav, Kimia. They're the one mostly providing all the device and the wetware that I've been using. And Fritjof, Ivan, they helped me construct a lot of pipelines and the supercomputer that I've been using to process the data. We share a lot of ideas and thoughts. So I appreciate them. And of course, I appreciate my advisor, Mattia Gazzola, for helping these projects. Okay, I can take any questions.
Cool. Thanks for the talk. I have one question. So these neurons are all excitatory neurons, right?
Yeah.
Do you have any idea why it doesn't sort of explode? Like, what's the mechanism that actually, makes these cultures sometimes show burst activity, but then also calm down again? Is that like just the refractory dynamics, or?
Yes, we don't have a good answer to that. And surprisingly, it was a very surprising for even Ivan when he saw this result, that they form avalanche. Because typically, in theory, without the inhibitory neuron, it's hard for them to exactly gather and form the avalanche behavior. The way we see it is because we see a lot of this like refractory behavior, and somehow with the combination of this like trying to blow up versus like a large refractory, kind of synchronized together and form this sort of a burst shape. So yeah, that's sort of our understanding at this point. We don't exactly have a good description of how this is actually happening. But yeah,
but how do you know that there's no inhibitory neurons?
On my end, because biologists told me, but they also told me that they have went through some staining method and they saw very low populations of the inhibitory neurons. So yes,
these are the stem cell derived neurons, or are these the stem cell derived neurons, right?
Yes, these are stem cell derived neurons.
Okay. So and the other thing is, when you showed the reservoir the criteria that it gradually dies out with time, what were you calculating exactly? After training?
After training?
Yeah, when you showed several plots with different frequency, different training, and then you showed maybe one before. Let's see. Can you go back one slide?
So during the testing period, we
Mhm.
tried to shine the pattern in the random sequence of input, and then we're trying to reconstruct back exactly the same sequence of input.
Okay, and you see how long does it take to do the same sequence back, right?
Yes.
So when you train pulse 1 second, one pulse every 1 second, and that's one training, is that right?
We train it for 1 hour. So yeah, we give a random sequence of the pattern for 1 hour. And then during the testing, we test it for like 3 minutes. So we gather about 180 samples to get the statistics.
I see. So when you say random training, mean in sometimes you are giving one pulse in 1 second. Some other time you are giving 5 pulse in 1 second. Is that right?
Yes. Yes.
And then when you test it, do you provide any signal like one pulse, 1 second, and see what is the response?
Yes, the type of encoding is the same. So yeah, we give like either 1 pulse or 2 pulse in a second. But the sequence would be different than the training. Yeah.
And then how do you compare? Is it like how do you say that it's fully recovering the trained output?
So
Is it
during the
cross-
training, during the training, we can learn the classification model. And then we're assessing whether this model is still valid after some amount of resting. So during the testing, so after we give this sequence of input, we're trying to see if our model is still valid to recognize the difference in the sequence.
I see. So when you showed that it's decaying with time, I think it's a slide before or maybe after, I don't remember now. Can you go to the next slide? Yeah, this is the one I'm talking about. So you're training 1 hour. That's the left plot, right?
Training 1 hour is this white block. Oops. My mouse. Yeah. So this is all the test results after the training.
After the training, okay.
So after the training, each of this dot is about 3 minutes of testing with the random sequence after this amount of rest. And then we are still assessing if the learned model is still valid for recognizing the patterns.
I see. So when you say RC training research study, so what is the y-axis? Reconstruction accuracy. What do you actually compute? How do you calculate reconstruction accuracy?
So there's 10 different patterns. And we can see how many patterns are recognized correctly and divided by the total number of patterns. That would be the accuracy.
I see. So you train first for 1 hour, and then you have a reset time of 30 minutes, right?
Yes.
What's a reset time? Okay, and then rest time of 30 minutes, and then you shine a signal like 1 hertz, like one pulse in 1 second, and then what do you read out? You read out what gives you the values of the what gives you the values, right? Okay.
Yeah, so the readout happens with the electrode, and we decode the state vector with the firing rate for each channel. And with this channel, we can run the classification. So if it's going to have like some firing behavior for different channels, we can test if that firing pattern actually means like which patterns. And then that's what the classification model is doing. And then with some random sequence of this cycles, we can measure the accuracy.
I see. So essentially you follow the sequence of firing, meaning which electrode fires after whom, is that right?
Sorry,
So when you have the RC map,
Mhm.
do you have to have some additional computation or something that you need to do from the readouts? So you have a large number of electrodes readouts.
Mhm.
Do you put some weighting functions, for example? Or no weighting functions here?
So
In-
after we read the firing rate for each of the patterns, so we'll have a very large, like a rectangular matrix of these patterns.
Yeah.
Then we can do the regression to get this linear map. And each of this map will tell us like which pattern for each row of the information is going to be. And our work is that after some amount of rest, this culture will change. This culture will evolve in some different dynamics, right?
Right.
I want to see if this learned map is still valid after that changes happen.
Okay. In that linear mapping, is there a weighting function that you have to evaluate?
This linear map is the weight function.
Yeah, so that's
needs to
what
be
I'm talking
searched
about.
for. Yeah.
Right. So that's the weighting function that you have to calculate, right?
Yeah.
I said that's the change. That is the one that is changing with time. So does the weighting function change with time?
No, this will be just trained only once at the end of the training,
Uh-huh.
and it's going to be fixed during the test.
I see. And then like every half an hour, is it that linear map changes because the neurons are evolving in some way?
No, we fixed the linear map because this is the external map that we just use. But the neural dynamic will change.
Dynamic, yeah. I see. So eventually you are characterizing the system, right?
Yes.
So I'm trying to understand how would you use actually to compute in whatever sense the computation be?
So here the computation can be anything. Like here I'm using the reconstruction just for the simplest like demonstration. So usually like computation would be like, if you give some signal of some continuous signal, we want to get some like a low pass filter signal, right? So then what this weight matrix will be doing is finding some weight that will compute out the low pass filter, right? That would be the form of a computing in the continuous signal using the reservoir computing framework. But here, like I'm just doing the reconstruction just for the simple demonstration, and because right now, like we don't want to make the problem like too complicated, because we want to understand like what is, how do we actually handle the system more than just dealing with the complex problem here.
Okay.
Other question, comments?
So is the control feed forward essentially not, but could you envision doing feedback control?
Yes, right now it's feed forward. We do have strategy for feed backward, which I'm testing like now, like right now. So yeah, that is somewhat down the line. So just like a pacemaker for the heart, there's a way to detect some abnormality in the frequency, and then you can specifically target perturbation to correct those abnormality. So those are some feedback algorithm that we're trying to test and design. We're trying to see if it works. Is there any more question on the Zoom or?
Do you know of any other RC computing system that are being used? Not neuronal, not living neurons, but where a dynamical system is used to compute something.
Yes, actually, there's a lot of people make a lot of silicon-based spiking system to do something similar. The idea originally started from like a liquid state machine where like people try to compute with like a water or some viscous fluid. So yeah, any physical system can actually replace the reservoir here. And also there's a lot of just theoretical model for the reservoir as well that people have been trying. So yeah, there are a lot of other works going on trying to see if they can use any other physical system to use it as a reservoir here.